For how long has this region been a desert? Was there vegetation here in the past? If so, how much vegetation and what kind? The answers to these questions may lie in the stable carbon isotopic composition of cave mineral deposits called speleothems. However, our collective understanding of how to interpret speleothem-derived carbon isotopic values is lacking and needs to be significantly improved in order to make reliable conclusions. Moreover, I'm investigating how to more reliably interpret these values by further constraining major carbon sources of speleothems. Since the carbon source of carbon dioxide in cave air directly reflects the carbon source of speleothems, knowing the major cave air CO2 sources will help to improve our understanding of what the speleothem carbon isotopic values represent. There is a consensus that atmospheric air is one of the major cave air CO2 sources, and it is thought that soil respiration is another major source. However, the hypothesis of my research project is that respiration in the bedrock fracture network rather than soil is the other major source of CO2 in cave air. Although both of these potential sources are from respiration, it is important to distinguish between these two CO2 reservoirs because the gas transport mechanism differs between the two matrices, and this affects the resulting isotopic values. If cave air CO2 is sourced from soil, then the gas would have undergone diffusion, a process that alters both the isotopic and molecular ratios of gases, as is indicated by the arrow length in the figure of diffusion on the left. On the other hand, if cave air CO2 is sourced from the bedrock fracture network, then the gas would be dominated by homogeneous pressure-driven flow rather than diffusion, and the isotopic and molecular ratios would therefore have not been altered since respiration. In order to solve this puzzle, I've collected cave air and soil gas from 2 caves in central Texas on a monthly basis and have compared both the isotopic and molecular ratios between the cave air and soil gas to see whether or not they display a diffusive signature. Based only on theoretical calculations, this plot shows the expected trends for respiration followed by diffusion in the solid upper line and respiration with no subsequent diffusion in the dashed lower line. Here the y-axis represents amount of oxygen and the x-axis represents amount of carbon dioxide, both with respect to atmospheric air, which is shown at coordinates 0, 0. You can see here that the soil gas data points plot along the expected trend for respiration followed by diffusion. This makes sense since we know that soil gas undergoes diffusion. You can see that natural bridged cave air data points clearly plot along the expected trend for respiration with no subsequent diffusion, and that inner space even plots below this trend, farther from the diffusive signature, likely due to calcite dissolution. If the cave air has not undergone diffusion as this figure suggests, then the dominant source of cave air cannot be soil respiration, but rather appears to be respiration from the bedrock fractures. However, our stable carbon isotopic data do not seem to align with this conclusion. The isotopic values indicate a soil respiration source in cave air, whereas the molecular values indicate a non-diffusive source of respiration. In order to resolve this discrepancy, we plan to further investigate carbon sources and transport. Once speleothem-derived stable carbon isotopic values can be reliably interpreted, their interpretations can be coupled with those of oxygen isotopes in order to explore causes for vegetative shifts through time that may be related to climatic shifts. These results can be used as an input to environmental models in order to project what the future may hold.